Okay, so we'll be looking at our outline involves introduction to the structure, function, nomenclature of the MHC molecule. We look at the clinical significance of the MHC molecule, also called HLA in humans, and its role in transplantation, transfusion, therapy, and disease. We are going to look at how to cross-match or determine the compatibility during tissue transplants, the methods that are involved in the cross-matching and the typing of tissue, although in introductory aspects of the procedures, method and procedures for HLA typing and cross-matching. We are going to mention them and define them and look at their advantage and disadvantage over each other. So our learning objectives, we expect that at the end of this lecture, you should be able to understand and explain what the HLA system is. Have a basic understanding of the organization and inheritance of the MHC complex. Describe the clinical significance of the MHC or HLA complex. Give an overview of the pre-transplantation tests and understand their significance. Demonstrates an understanding of the principles behind the molecular techniques used in HLA typing. Okay. So the major histocompatibility complex. We are going to look at some definitions and take the introductory aspects. Okay, so the major histocompatibility complex. This is the peptide display system of adaptive immunity. Its function is to display peptide fragments of protein antigen for recognition by antigen-specific T cells. In humans, the MHC is known as the human leukocyte antigen. The MHC complex in human, they are known as the HLA antigens because they were discovered on human leukocytes. So it's a system, an adaptive display system, adaptive immunity, where the body processes antigens and presents them on this complex for recognition, okay, for recognition and destruction by the adaptive immune system of the body. So it's a system that works with the adaptive immune system of the body, where the antigen that we are processed are presented on this complex for recognition and destruction, okay, by the T cells. So that is what this definition is trying to highlight, and that in humans they are referred to as the human leukocyte antigen because they were discovered on human leukocytes, that is the white blood cells. So the MHC complex was recognized as a genetic complex that influences the ability of an organism to accept or reject transplanted tissue. The particular set of MHC molecule a certain individual expresses determines which antigen that person's T helper cells or T cytotoxic cells can respond to. As we go along, you'll be understanding each of this definition of the MHC complex. The MHC complex play three major important roles. They are very, very important in antigen presentation, autoimmune disease, and in transplantation. So this complex of the human body was recognized during the early work of Goa and as antigen that are responsible for allograft rejection in mice. Okay, so they discovered that in those time during tissue transplantation, some tissue transplants are successful while others are not successful. So they tried, because the tissues of the body are involved, they named it histocompatibility complex. Okay, the development of congenic and recombinant strains of mice by Snell enabled the detailed analysis of various loci of this complex. But the foundation of the HLA complex were laid down in 1958 by three scientists, Jan Dorset, Jan van Rode, and Rose Pine. They described the presence of alloantigens on leukocytes in human sera, obtained from multiparous women or patients who had received multiple transfusions in three separate papers. Okay, so with the discovery of alloantigens on white blood cells on leukocytes in humans, okay, this complex was named the human leukocyte antigens in human beings. So among the three, they were given Nobel Prize. Jane Dorset was given Nobel Prize. The discovery of the HLA, okay, they discovered the human leukocyte, the alloantigens on leukocytes of pregnant women that have been pregnant previously, or people that have received multiple blood transfusion. They observed these alloantibodies, alloantigens on leukocytes, on their leukocytes. Okay, so because of the immune reaction that are involved during pregnancy and also during blood transfusion, these scientists observed that the human leukocytes of these persons that have been pregnant over sometimes, that's multiparous, that is multiple pregnancies, okay, they observed that they've developed alloleukocyte antigens. They recognized, they discovered these alloleukocyte antigens, alloantigens on their leukocytes, okay, because of the fact that they have been exposed to some immunological responses of pregnancy and multiple blood transfusion. So they refer to this complex as the human leukocyte antigens. Okay. The discovery of the HLA is the first milestone in the field of immunohistocompatibility. However, the impact of immunohistocompatibility testing in transplantation was established in 1969. Petel and Teraska demonstrated in kidney transplantation recipients and donor pairs that there is a strong correlation between a positive lymphocyte cross-match and a hyperacute rejection as a common outcome. So they continued to investigate and discover a lot of immune reactions. For example, during kidney transplantations, they observed that persons with high positive leukocyte cross-match, the tendency of having tissue rejection, that is rejection of hyperacute rejection of a transplant, is possible. This compelling data resulted in a mandatory prospective cross-match before kidney transplantation to identify the antibodies, that are involved. To identify the antibodies that are involved. Sorry for that network interruption. Okay, so the discovery of the MHC complex started first with the discovery of tissue rejection during tissue transplants in animals like mouse. So it was also discovered in humans, especially in multiparous pregnant women and those that have received multiple blood transfusion, that there were allo antigens to human leukocytes these persons developed as a result of pregnancy and blood transfusion. So they now also observed lymphocyte, the reaction of they cross-matched the lymphocyte and found out that those with strong correlation, there's a strong correlation between a positive lymphocyte cross-match and a hyperacute rejection. Okay, so these are some of the early studies that led to the discovery or development of this MHC complex. So we're going to look at the genetics. So MHC complex was named so because the molecule played a crucial role in tissue transplantation. In both humans and mice, MHC genes are arranged into regions encoding 3 classes of molecules. We have the class 1 MHC molecule genes. This is expressed on surface of almost all nucleated cells. Major function is presentation to T cytotoxic cells, present to intracellular pathogens and peptides. We'll see all this in details. Let's take it gradually. Okay, we have the class 2 MHC gene. They are expressed primarily on antigen presenting cells, the macrophages, dendritic cells, the B cells. Their major function is presentation of antigen to T helper cells. Okay, they present extracellular pathogen peptides like bacteria. You know, when some pathogenic condition like HIV, you know, HIV, during HIV infection, the virus invades the cell. Okay, the HIV virus invades the cell and takes up the cell mechanism. So for the body to mount an immune response to this HIV invasion, it's involved the MHC class 1 genes. These genes are expressed on the surface of almost all nucleated cells, and their major function is presenting to cytotoxic T cells. Cytotoxic T cells, you know, they will destroy the infected cells. So because they will process the virus that have infiltrated, gain access into this inside of the cell, they process intracellular pathogens, that is pathogens that have invaded inside of the cell. These MHC molecules class 1 are involved in processing such pathogens, and they present it to the cytotoxic T cells, which will now destroy those infected cells. But in the case of the class 2 MHC molecule, they process and present extracellular pathogens, maybe like bacteria rolling around side of the body plasma. They process it and then they present to T helper cells. You will understand it gradually. Just take it gradual. So class 3 MHC gene, they encode for secreted products that have immune functions. That is components of the complement C4, C2 factor B, and tissue necrosis factor. So classification, chromosomal location. You can see this chromosome number 6, the MHC molecule is located at this region of the chromosomes, chromosome number 6, at the short arm of chromosome number 6. You can see this region. This is where the MHC molecules is being housed. Okay, so and this molecule have the class 1, have the class 2, and then we have the class 3. But we are going to discuss only the class 1 and the class 2. The class 3 only codes for secretory proteins. Okay. So when you look at this in-depth structure of the MHC region on chromosome 6, it contains two sets of genes encoding for the MHC class 1 and class 2 molecules. You can see the class 2 molecules, okay, DP, DQ, DR, DM. Can see the class 1 molecule A, B, C. Okay, this is the genetic mapping for this MHC molecule. The class 3 locus contain genes for complement protein and some cytokines. So the HLA complex is encoded on chromosome 6. It contains more than 200 genes and extend over at least 4 million base pairs. Okay, class 1 HLA molecules, called the alpha chain molecules, is encoded by A, B, and C loci in humans. The beta 2 microglobulin chain, when we go to the structure of the MHC molecule, you'll be seeing these chains of the MHC molecules. The beta 2 microglobulin chain encoded by a gene outside the MHC and is located on chromosome 5. So there is this chain, beta 2 microglobulin, that is in the class 1 HLA molecules. So it's we are to note that its chromosome is located in chromosome 15 and not 6 of the MHC molecule generally, okay, and that encoded by a gene outside the MHC complex. It's good to note that, although it's part of the chain of the class 1 HLA complex, but it's being encoded by a different chromosome outside the MHC complex. There are also non-classical class 1 molecules. We have others that are not classically class 1 molecule that also encoded by additional gene or groups of gene within the class 1 region. Then the class 2 molecules are encoded by the DP, DQ, and DR regions for both the alpha chain and the beta chain. Okay, for each HLA class 2 isotype, the gene encoding the alpha and beta chain are called A and B respectively, like HLA DQA, HLA DQB. These are the genetic terminologies used to name the HLA complex. When there is more than one gene, including non-functional gene, a number in series is added. We'll see all these things later. You will understand it later, but just keep following me. Okay. Other non-classical class 2 molecules these are also encoded within this region. Okay. So when you look at this this slide, you can see how it's trying to show how the MHC molecule is being named. Okay, so it's a very complex group. It's a very complex group. The naming follows some laid down rules, like the first letter denotes the HLA, then this alphabet shows the gene. Okay, then this next field 1, indicates the allele group. The field 2 indicate the specific HLA protein. Okay, field 3, okay, synonymous DNA substitution encoding region. Field 4 is showing us changes in non-coding region. Then this suffix This alphabet denotes changes in expression. Like N shows that it is a null allele that is not being expressed. If it is L that is here, it shows a low level expression. If it is A that is here, it would mean aberrant expression. C, molecule present in cytoplasm. If it is S, means that it is secreted molecule present only as a soluble form. So this is, like, call it the IUPAC nomenclature. That's the something that you can easily use to, that is similar to, this is the IUPAC nomenclature for the MHC complex. That is what you can use to really know what is happening here. So that's all. So the allelic forms of the MHC gene, that is the haplotypes in humans, we are going to discuss the inheritance. In an outbreed population such as human, each individual is usually heterozygous at each locus. Okay, you are usually heterozygous because you inherit from your parents. Okay, a haplotype is a set of alleles inherited from one parent as a set. Possible haplotypes, we don't go into details into this. Okay, so we also discovered that the MHC is has very diverse. It shows a lot of diversity in its nomenclature or in its function or how it operates or how it operates. Okay, so we've said that it's located on the genes. We have the chromosomes. We have the genes for class 1, class 2, and class 3. Then the locus, we identify the locus for both the class 1 and then the class 2. And then the alleles, 2 or more alternative forms of a gene at a particular locus that confer alternative characters or that code for slightly different varieties of the same product. These are some of the terminologies you'll be coming across. Okay, the MHC system is polymorphic because at each location there is a multiple possible allele arrangement. Loci constituting the MHC are highly polymorphic. Genes for MHC lie close together. Recombination frequency is very low, hence rare crossing over of events. So these are some of the genetic characteristics of this MHC complex. It's highly polymorphic because there are so many alternate forms of the same gene, which we'll also discuss in details as we proceed. So the MHC allele, the arrangement or the inheritance, each set of allele is referred to as a haplotype. Individuals inherit one allele from each parent. Heterozygous, different alleles at one locus, more diverse, okay? Homozygous, when someone inherits the same alleles from father and mother. Okay, these alleles are codominantly expressed. Both maternal and paternal gene products are expressed in the same cell. Heterozygous, inheriting the same allele from both parents results in a person being homozygous for that one MHC allele. Okay, so what we're trying to summarize here is that the MHC complex, the inheritance of MHC complex is codominant. Any allele you inherit from your mother and the one you inherit from your father are expressed. Okay, there is no um, they are codominantly expressed. There is no recessiveness in MHC complex. Whatever you inherit from your father or your mother, you express it. That is, it's functional in your system, in the same cells. Okay. MHC type is unique, like a fingerprint, that is making it very unique. like a fingerprint. No two persons are the same. It's this principle or these characteristics or this property of the MHC molecule can be of an advantage or of a disadvantage. Okay, we'll see that later. It can be of advantage because of the fact that it is like fingerprint, that is, no two individuals are the same. That is the complex. It helps in paternity disputes. So when they are doing paternity disputes, they are actually analyzing the MHC complex. Okay, that is the good parts of its polymorphism, okay, or its diversity. That is, it's also good in the sense that it also helps population to resist epidemic. Okay, because of its diversity. This is another advantage of the MHC molecule. We mentioned that it helps in paternity dispute testing. Okay, it also helps a population to be able to mount and resist any epidemic. In the event of any epidemic, because of its diversity, because of its ability to exist in different forms, okay, no matter the type of new pathogen that is invading the population, okay, the population will be able to mount an immune response because it has a variety, diversity of mechanism to do that. That is on the good side of the MHC molecule. But one major disadvantage of the MHC molecule is in the problem it creates or it generates during the process of tissue transplantation. Take, for example, during the process of kidney transplant. You find out that it makes it difficult to get a 100% match for someone for kidney transplant. And that is what necessitates the immune suppression that is involved before and even after the transplant, so that the body does not mount an immune response against the transfused organ. Okay, so the treatment, part of the treatment or management of kidney or this transplantation is suppressing the immune system of the person and predisposing them, okay, to opportunistic infections. So it has its advantage, it has its disadvantages. So, like we said, one of the main characteristics of the HLA is its extreme polymorphism, ability to do different forms. Among the expressed loci, the HLA has the greatest degree of polymorphism in the humans. Okay, it has the greatest degree of polymorphism. Okay, different forms in humans, in human genome. The number of known alleles increase constantly. Okay, you can see the number of alleles that are being discovered in this complex is increasing every day. Okay, I've not even updated this. This is at 2023. If you search now to get the 2024 HLA alleles that have been discovered, you find out that it keeps increasing because it is a gene, it is a complex that is highly polymorphic. It exists in different forms. Okay. So expression and classification of the MHC gene. So the MHC gene, we said we have class 1, class 2, class 3, but our concern, our main discussion will zero in on class 1 and class 2. So this class 1 MHC complex, we say that it is located in all nucleated cells. Now you ask yourself, which cells of the body are nucleated and which ones are non-nucleated? you find out that all the white blood cells, mature cells, are all nucleated. But you know that the mature red blood cells does not have nucleus. So the mature red blood cells does not express HLA class 1. So what is the function of this HLA class 1? Its function is in recognition of tumor and virus infected cells. Okay, it will recognize tumor and virus infected cells because it processes intracellular pathogens like viruses that have invaded inside of the cell, taking up the cell mechanism. So to destroy these viral infected cells, the part of the adaptive immunity of the body that is engaged is the HLA class 1. It is engaged because it will be able to recognize this intracellular pathogen, process them, and then present them to the cytotoxic T cells to destroy the invaded cell. Okay, now the class 2 MHC complex, we've said that the MHC molecule are involved in adaptive immune response of the body. Okay, so the class 2 MHC molecules, their location, we can find them on the antigen presenting cells. What are the antigen presenting cells of the body? We have the B lymphocytes, we have the macrophages, we have the dendritic cells, endothelial cells. Now, what is their function? They recognize tumor and virus infected cells by the CD4 TL pathogenic cells. They recognize extracellular pathogens. Okay, they recognize extracellular pathogens, process it and present it to T helper cells. There's some mix up here. They recognize extracellular pathogens and present it to T helper cells. Okay, these are the genetic mappings for the HLA complex. This is the HLA complex in humans. This is what its counterparts or its analog in the mouse. Okay, we are talking about humans. Okay, just like I said earlier, what's the clinical significance of the HLA complex? The transfusion or transplantation of blood components bearing allogenic HLA can induce clinical significant immunological responses. You have to recall our transfusion reactions we did in fourth year. You have to recall that class because in that class we discussed that although we do cross-matching in the blood bank, majority during blood transfusion, what we do majorly is we are trying to cross-match the red cell antigens and the antibodies. During the process of blood transfusion, you know that blood transfusion is a form of tissue transplant, and that during this process, what we majorly do is to do what? Cross-match the red cell antigens and the antibodies. We don't take into account cross-matching for white cell antigens. And we are saying here that, and we are saying here that alloantigens of leukocytes have been recognized in those that have been multiply transfused, showing that there are reactions between the leukocyte antigens of different individuals during that process of blood transfusion or as a result of multiple pregnancies. Okay, we have recognized that there are antigens too. white blood cells, not only antigens to red blood cell. So in the process of blood cross-matching, blood transfusion, we are silent about the white blood cell antigens. We worked only on red blood cell antigen and antibody, trying to make sure that they match before we transfuse. But we are saying now that this discovery of the HLA antigen has shown us that the leukocyte, the white cells, also have antigens which should be cross-matched, but we don't do that. So it poses some problem. It poses some problem during the process of during blood transfusion, and that is why before someone is being transfused, you find the clinicians giving antipyretics against fever. Okay, so the transfusion or transplantation of blood components bearing allogenic HLA can induce clinically significant immunological responses. Go back and read your blood transfusion reactions. Look at the transfusion reactions that are being triggered by leukocyte antigens, antibodies. You get what we are saying correctly. So all cellular components except the erythrocytes express HLA, okay? And any plasma containing products may include HLA specific, the antibodies may be there, okay, which are potentially harmful to the recipient. So this is what causes transfusion, some of the transfusion reaction we discussed in our 422, which you have to go back to see. So the MHC class 1, we are still trying to lay background on this complex for you to understand what the complex is all about, how it contributes to the body immune system, and its advantage and disadvantages. So the MHC class 1, we say it's expressed in most nucleated cells. Different cell types lead to different level of expression. The highest expression is on lymphocytes. Okay, so go back to your transfusion reactions and check the transfusion reactions that are being caused by these cells. The lowest expression on fibroblasts, muscles, liver, and neuronal cells. The highest expression is on the lymphocytes. Cells that lack MHC1 neuron, like sperm cells, RBC cells. Normal cell will display cell peptides from normal turnover of cell proteins and viral peptides. Okay, each MHC can present different peptides from the same virus. MHC class 1 present endogenous peptides. I've been hammering on these things. Endogenous peptides, that is, originate from inside the cell. And I've given you example of the viral cells, that the HIV virus cell that can invade the cell mechanism and take over the machinery of the cells, that in the adaptive immune response of the body, that the MHC complex that is being engaged to handle it is the MHC class 1, because it will be able to identify these intracellular pathogens, okay, process them and present them to the cytotoxic T cells for destruction. I hope that is very, very clear. Okay. So we are going to look at, am I sure I'm right? Okay, still in order. Okay, so we're going to look at the structures of the MHC class 1. When you look at this diagram here, you find out that this chain is a continuous chain. You can see this is one chain. This is one chain. It's continuous, PAM, then enters inside the cell. Then this is another chain. Okay, you can see how the class 1 is. The groove is formed by the alpha 1 and alpha 2 chain. Okay, so this is the alpha chain. See how it goes like this, it goes continuous. The alpha chain that form the groove. The beta chain, we said it's been encoded by another gene that is outside of the MHC, another chromosome that is outside of the MHC complex, that is chromosome number 15. And you can see that in its structure. This is the beta 2 microglobulin chain of the MHC class 1 chain. You can see it here, that this chain that is being encoded by a different chromosome outside the MHC complex. Then this alpha chain is being encoded by the chromosome number 6, I think so. So you can see how the structure goes. So the MHC, that is the important thing. And then when you look at this is the surface where the antigen presentation occurs. Okay, the MHC class 1 molecule processes intracellular pathogen and presents it at this peptide binding clef. Okay, when it presents it, it triggers off the cytotoxic T cells to come along and then destroy the cell that is involved. Okay, hope it's becoming clearer to you. All right, so the MHC class 1 molecule are heterodimers. 45 kilodaltons alpha chain. Okay, 45 kilodalton alpha chain, I've shown you that, formed with 3 protein associated non-covalently with a 12 kilodalton beta 2 microglobulin. This is it here. Okay, the alpha chain is a transmembrane glycoprotein encoded by A, B, and C region. The alpha chain is transmembrane, passes through the membrane to enter inside of the cell, is transmembrane. Okay, the beta microglobulin is encoded on a highly conserved gene on a different chromosome 15. We said that earlier. The beta 2 microglobulin the beta 2 microglobulin is used as a tumor marker, for example, a multiple myeloma. Increased level of the beta 2 microglobulin shows a poor prognosis of the tumor. The alpha 1 and alpha 2 domain interact from a deep groove which can bind a peptide that is 8 to 10 amino acid long. So this groove, this alpha 1 and alpha 2 chain, alpha 2 groove of this alpha chain forms the groove or position where the peptides are being bound, and it can bind up to 8 to 10 amino acids. Okay, the alpha 3 region, you can see the alpha 3 region here, okay, is highly conserved and interacts with CD8 on T cells and beta microglobulin. Who is shouting there? Okay, an alpha chain to undergo proper folding and transport to the cell surface. Wow, why are you shouting? Okay, so for the class 2 MHC molecules, okay, they are only expressed constitutively by antigen presenting cells against class 1 molecule where we said that they are being presented by all nucleated cells. But the class 2 molecules, MHC molecule, are expressed only by antigen presenting cells. Mark those differences on your brain. Professional antigen presenting cells, we have the B cells, the macrophages, and the dendritic cells. Non-professional antigen presenting cells, we have the epithelial cells like the thymus, thyroid, fibroblasts, glial cells, pancreatic beta cells, vascular endothelial cells. Level of expression varies at different stages of maturation or activation. MHC class 2 is not detected on pre-B cell, but is constitutively expressed on mature B cells. MHC is upregulated on activated monocytes and macrophages. MHC contains 2 different polypeptide chains, alpha and beta. Who is raising up with your hand here? Anayo, You have any question? What's your question? Unmute yourself. Let me see if we can take it. Our time is fast moving.
Sorry,
You're
ma, it
raising
was a
up.
mistake.
Oh, yeah, put down your hand now. All right. So let's go ahead. We'll soon round up. Okay. So the structure of the class 2 molecule, I'm just taking it slowly so that you get one or two things, because this is not our normal hematology red blood cell stuff. Okay. So the MHC class 2 molecule, can see the structure here. When you look at this structure, it's different from the class 1 MHC structure. You find that it's made up of the alpha chain, which is on its own bar, goes this way, and then intracellular too. Then you have the beta chain here. It goes this way also, but also crosses inside the cell. So what it means is that this groove binding, peptide binding cleft, is being formed by the alpha 1 and beta 1 chain, unlike the MHC class 1 structure. So you should be able to differentiate the structure of these two molecules. The MHC2 contains two different chains, 33 kilodalton alpha chain and 28 kilodalton beta chain. The alpha 1 chain and beta 1 chain form the cleft where processed antigen binds. Note that beta 2 is not beta 2 microglobulin, as we said in class 1 MHC. This is not beta 2 microglobulin. Peptide cleft can bind up to 13 to 18 amino acid. The other one can bind what? 8 to it can bind 8 to 10 amino acid. Okay, while class 2 can bind 13 to 18. Okay, both the alpha chain and beta chain are made up of two domains. Both the alpha chain and beta chain are made up of two domains, alpha 1 and alpha 2, beta 1 and beta 2, as against MHC class 1 that has three domains for the alpha chain and one beta 2 microglobulin. When you go back there, you see it. Okay. All right, they are membrane bound. You can see they are membrane bound glycoprotein that contain external domains, a transmembrane agent, and a cytoplasmic tail. Okay. So what we take out from this introductory lecture is the factors influencing MHC diversity and immunity. We say that the MHC complex has is co-dominant, expression of both paternal and maternal genes. Okay, whatever you inherit from your father in the MHC complex and your mother in the MHC complex are being expressed on the same cells. There is no recessiveness on the MHC complex inheritance. Everything that are inherited are expressed, are dominant, and they are expressed. The haplotype, we talked about the haplotype. The inherited sets of alleles from each of the parents. Each closely linked set of alleles that are inherited is called a haplotype. Okay, we say that the MHC complex is highly polymorphic. That is the existence of many alternative genes and alternative gene forms within the population. Many alternative gene forms within the population, and we also explain that this has its advantage and its disadvantage. We also talk about the polygenic nature of the MHC complex. Multiple genes with similar function result in tremendous diversity of the HLA molecule and T-cell immunity within human population, helping them survive epidemic disease, but also being the main immunological barrier to clinical organ transplants. tight linkage, close proximity of gene leading to inheritance patterns. The recombination frequency within this gene is very small. Hence, allele encoded by this loci are inherited are two sets, one from each parent. What are these people? One from each parent. Okay, so I think we'll call it a day today. This is just an introductory part of it. The next lecture, I think we are going to be going into the antigen processing and presentation, how the MHC class 1 processes antigen and presents it, how the MHC class 2 process antigen and present it to the adaptive immune system of the body. So that will be our next lecture. For this, we did introductory aspects to the MHC complex, and we said it's part of the body's adaptive immune response. Okay, and we're able to describe its function. We'll be able to describe the classes and the properties, the characteristics of the properties of different classes. And that's all for now. So if you have any questions, you go ahead and then ask your questions now.